polyploid, all kinds of strange things. They turn sexual reproduction upside down. They reverse evolutionary trends. They take the initiative in evolution. In a way, of course, that the Darwinians and the neo-Darwinians and Morgan himself, of course, had no conception of and didn't want to have any conception of. It was something that you could not predict without a long-term, painful, arduous study of the chromosomes themselves. Now we know, therefore, that the chromosomes trip and trap the hereditary and evolutionary processes again and again in ways that experimental breeding cannot easily reveal to you. We know that they give us a whole world of new ideas, both of heredity and of evolution. And we know, in short, that the chromosomes are behaving in a certain sense as organisms in their own right. Don't misunderstand me. I'm not trying to cavil at the reductionist principles that have taken us so far. I've been an adherent of those reductionist principles for 50 years. I don't hesitate in sticking to them, but all the same, there is this big element in between. These chromosomes here are very big and very important, and they stand between the genes and the chromosomes, and they can never be omitted from our studies of heredity or of evolution. Now, this is true in the first place at the microscopic level. But of course, now, today, we can see here, every day more, that when we look at the chromosomes at the molecular level, even more so, is the initiative taken within the lives and within the processes of chromosome evolution. At the molecular level, if we think of it so, a new horizon altogether is opened up to us, which the Darwinians and the neo-Darwinians could never possibly have conceived. Thank you.
The next speaker is Professor Arne Münzing of Lund, whose introduction is entitled "The Importance of Chromosome Identification for the Solution of Some Problems in Plant Cytogenetics."
Mr. Chairman, ladies and gentlemen, I am very much afraid that after the brilliant introduction to this session by Dr. Darlington, what I have to tell you will seem very trivial. But to my excuse, I can perhaps tell you that the starting point of my information goes back more than half a century, but has a very clear connection with what is going on today. The starting point of my paper is sterility. More precisely, a striking difference in sterility between animals and plants. In animals, chromosome aberrations as well as lethal genes are always transmitted by the gametes, but may be removed by zygotic sterility. In higher plants, in which the haplophase is more differentiated, haplontic sterility may occur. This means that certain classes of pollen grains and embryo sacs are killed by their own genetic constitution. Haplontic sterility is differently expressed in self-fertilizing and cross-fertilizing plants. Strict self-fertilizers are completely homozygous, and consequently all their gametes have the same genotype. This genotype gives fully viable pollen grains and embryo sacs. Otherwise, the species in question would, of course, not be able to exist and would very soon disappear. However, if different biotypes of the same self-fertilizing species are intercrossed, the F1 hybrids may sometimes be partially sterile. This is due to elimination of certain classes of gametes. Such is the situation in Galliopsis tetrahete, a very polymorphic weed, which was my favorite material in the 1920s and 1930s. In a certain cross involving dihybrid segregation of flower color, the two recombination gametes were entirely eliminated in the pollen and partially eliminated in the ovules. This, of course, disturbed the segregation ratio in a characteristic way, which was analyzed in detail. The main result was that we had here a clear case of haplontic sterility. Certain categories of pollen grains were entirely killed by their own genotypes, while pollen grain with other genes were perfectly normal. On the female side, lethality was less pronounced. Those genotypes which always killed their pollen grains were sometimes transmitted by the ovules to next generation. In the crosses between pure lines of Galliopsis tetrahete, the F1 combinations were characterized by various degrees of fertility. Some combinations were apparently perfectly fertile, others were more or less pollen sterile, with the corresponding reduction in the degree of seed setting. A typical example of a cross-fertilizer is rye, Secala cereale. In this species, the pollen of 600 individuals was examined. About half of the plants were really found to be partially sterile, with a very wide range of variation. In most of the other diploid cross-fertilizers, the same picture was repeated. Thus, in diploid cross-fertilizers, partial pollen sterility seems to be a normally occurring phenomenon. Detailed work concerning the nature and inheritance of partial sterility in rye was carried out. The general conclusion was that population plants of rye are genotypically different with regard to the factors causing various degrees of partial sterility. To gather some information about the true nature of these sterility factors, the mode of meiosis was studied in two different rye varieties. The idea of a specific normal standard set of chromosomes in rye and other cross-fertilizing organisms, including man, may imply a certain degree of schematization. Probably there is no definite and clear-cut standard type of chromosome structure. On the contrary, the chromosome complements in the gametes are frequently different in several structural details. Sometimes the differences carried by the two gametes forming a new individual are large enough to give rise to multiple chromosome associations or other conspicuous deviations. In most cases, however, the differences are too slight to influence meiotic chromosome pairing. In recent years, the GIMSA technique has not only made it possible to identify the 7 different rye chromosomes. The bending technique has also more accurately than before revealed structural chromosome differences between species of rye and between different varieties of cultivated rye. In 1975, Singh and Röbelin in Germany and Weimark in Sweden also reported cases of heteromorphic chromosome pairs in various types of cultivated rye. Weimark also observed heterozygosity for bands in population plants of a variety steel rye, and in inbred lines of the same cultivar, she got evidence of homozygosity for larger structural deviations including reciprocal translocation and pericentric inversion. Such data indicate the difficulty of defining a perfect standard karyotype in Rye, and also fit in very well with the previous conclusions from the old work in Galeopsis and Rye. In some cases, there is reason to conclude that not entire chromosomes, but only chromosome segments have been substituted. Precise information will probably soon be achieved with the aid of modern banding technique. Consequently, this technique is now also invading the plant kingdom, almost explosively. Thank you.
Our next speaker is Professor Torbjörn Kaspersson of Stockholm, whose title is Biophysical Techniques in Chromosome Analysis.
Mr. Chairman, may I thank for the opportunity to speak here tonight. In order to fit better in the plans of our chairman for this session, I will, with an apology, take the liberty to go somewhat beyond what's implied by precise wording in the print program and speak in somewhat more general ways about the biophysical techniques in chromosome analysis. The supplementation of conventional microscopy by different other physical techniques in the first-line measuring procedures proved to be all aiming at the use of the microscope as a quantitative tool, proved to be of some value for chromosome studies already quite long ago. In the 20s already came the quantitative polarization techniques, which found rather little application in biology. In the middle 1930s, the first ultramicrospectrophotometric procedures were developed, and during the 40s and the 50s they were joined by X-ray based measuring techniques and fluorometry and also microinterferometry. Amount and distribution of many different cell constituents can be determined with these techniques. For works on nuclear components, spectrophotometry and fluorometry have proved most useful. It was in fact the interest in the cytochemistry of chromosomes and chromatin which gave the most important stimuli in these early days to development of new cytochemical biophysical procedures. During the years of the Second World War, our group in Stockholm concentrated on the relation between RNA and protein synthesis, but shortly after the war, cytochemical chromosome work was again running full speed. We had the great privilege then of having as long-time staying guests such grand men of cytology as Lothar Geitler and Hans Bauer, and later on his pupil Wolfgang Biermann, all aiming at the application of biophysical techniques of the type I've indicated on chromosome problems of several different types. That was mainly absorbometry and also ultraviolet dichroism measurements. During the following decade, many laboratories worked cytophotometrically with DNA determinations. And the reason why DNA was so frightfully popular and still is that that is the by far most easy substance to determine cytophotometrically. And this work with running in many laboratories contributed, as you know, to the development of the idea of a DNA constancy. And later on also, it was a contribution to the development of our present views on the role and mode of function of the DNA. Little was done on the cytochemistry and structure of individual chromosomes during this rather long period in the 50s and early 60s. This was, however, also the time when cancer research expanded so fast all over the Western world, and that tended little by little to focus interest again on the behavior and on the structure of individual chromosomes. And now the emphasis was on, because of several reasons I can't go into, was on the mammalian chromosomes. Other inducements for research of that type, stimulating interest for chromosome studies, were the atomic bomb test in the 50s and in the early 60s, and also the coming of the atomic energy, as mirrored, for instance, in the United Nations activities in creating that UNSCARE committee, the scientific committee for the study of the effects of atomic radiation, in which studies, chromosome analysis and chromosome studies played a considerable role. If I look just at our own experience from the last few years, we find that we can easily gain at least one magnitude and possibly more in the speed of the aberration analysis in this way. And I cite that here in order to indicate that there is very much that one can get in this way, and I am sure that such techniques will be developed in the future very much, and still further facilitate the work and speed up the application of the magnificent many different techniques we have for aberration analysis. And these are good examples, I think, of the ever new possibilities for more penetrating chromosome analysis, which has been offered by biophysical quantitative cytochemical techniques during a period of several decades. Thank you.
Our next speaker is Professor Albert Levan of Lund, who will speak of the development of chromosome research in cancer.
Mr. Chairman, ladies and gentlemen, that malignant growth is associated with manifold cellular and nuclear abnormalities was well known to the pathologists of the 1890s. Their observations stimulated Boveri to propose in 1914 his somatic mutation theory for the origin of cancer. Thus, the cancer problem was early recognized as a concern of cell genetics and chromosome research. During the following 35 years, cytogeneticists did not pay much attention to chromosomes in malignant cells. There was one exception, Winge, the Danish cytologist, who in 1930 published an important paper on the chromosomes of experimentally induced tumors in mice.